Okay, everybody. We want to get started because we've got an extensive and exciting program for you this morning. So welcome, everybody. Thank you for coming. There may be one group who's a little bit delayed because of traffic, I think, but they'll just come in unobtrusively for the back. So I'm Ron Perlman, Dr. Perlman. I'm a professor in the Department of Biology here at York, and I'm also associated with the Gairdner Foundation, who are bringing this program to you today. So I just want to start, and I won't take too long, just a few minutes to tell you a little bit about the Gairdner Foundation, the group who's bringing this exciting event to you today, because I think most of you probably know very little, if anything, about the foundation, and I hope that'll change after today's program. So on behalf of Dr. John Dirks, the President and Scientific Director, and Dr. Lauren Terrell, the Chair of the Board of the Foundation, which is now in its 56th year, I am delighted to bring greetings from them and to welcome you to this event. The foundation was founded by a man by the name of James Arthur Gairdner in 1959, and he was convinced by friends and family to put his considerable wealth to good use, the recognition of excellence in scientific endeavor that leads to good for society. So the awards are supported by a recent significant endowment from the Government of Canada and now include what are called the Canada Gairdner International Awards, the Canada Gairdner Whiteman Award, and now entering its 6th year, the Canada Gairdner Global Health Award. Your speakers today, who will be formally introduced shortly, are a 2014 Canada Gairdner Whiteman Award laureate and a member of one of the Gairdner peer review committees called the Medical Review Panel. These awards are Canada's most prestigious awards in biomedical science and rank amongst the most prestigious scientific awards in the world. The Gairdner Awards are unique in many ways, including the national program that we are part of today. So this national program brings present and past award winners and members of what we call the Gairdner family to speak about their award-winning science at 22 venues across the country. And in 16 of these venues, such as here right now, we speak to young high school students like yourself. The program puts scientific excellence at the forefront of Canadian science. Gairdner events, a true trans-Canada celebration of scientific excellence, will culminate in Toronto next week with a series of exciting symposia, award presentations by the laureates, and a gala black tie awards dinner. We work our speakers and laureates hard, but they find it enjoyable and worthwhile. This presentation at York has been part of the Gairdner National Program for, I believe, 10 years now, and we're delighted to have the opportunity of partnering for this exciting program. The program for you today for high school students brings the excitement of scientific excellence to young students, the next generation of scientists, the future of scientific excellence. And the idea of this program is not to teach science, but to use award-winning science to talk more about the scientists, how they decided to go into science, who their mentors were, the excitement of doing science, and providing advice to students to excite the students about biomedical science. The Gairdner lectures, which we will hold later today, this afternoon, are research presentations to faculty and trainees. As some of you know, the Gairdner Awards, as well as being very prestigious, are also excellent predictors of Nobel Prizes. Nearly 30% of Gairdner Award winners have gone on to receive Nobel Prizes. Recent history speaks to an especially strong performance in the Gairdner Nobel continuum. In 2013, for example, 2 Nobel Prizes in relevant fields were awarded to previous Gairdner Award winners, and this year, one of the Nobel laureates in physiology or medicine was last year's Canada Gairdner Global Health laureate. In the 56 years of Gairdner's existence, now 83 Gairdner laureates have gone on to receive Nobel Prizes. Pretty phenomenal record, I think you'd all agree. Sponsors have been listed, they're listed there. We can't do these things without the support of the Government of Canada, Government of Ontario, Canadian Institutes of Health Research, the Globe and Mail as the media sponsors, and certainly our local organizers, York University, and particularly I'd like to mention Kate Duncan, who coordinates this program. She's in admissions and recruitment at York, and we graciously acknowledge and are grateful for all of the help. So it's my pleasure to welcome you here today on behalf of York and Gairdner, and to thank you for coming to this exciting program. I'm sure that you will enjoy today's program. And now I'd like to introduce you to Mr. Waqar Ahmad, an exceptional fourth-year honors student in York's biomedical science program, and he'll introduce the first speaker. Waqar.
Good morning, everyone. My name is Waqar Ahmed, and I'm a fourth-year biomedical science student here at York University. I'd first like to start by expressing my gratitude. I feel incredibly privileged to be a part of the proceedings of the 10th Annual Gairdner Awards, Canada's most prestigious biomedical science awards, here at York University. As you have heard, the Gairdner Awards recognize accomplished researchers from all over the world. Today, we'll hear from two trailblazers in research and their journey in science. But before we hear from them, I'd like to talk to you about you. As high school students, I know many of you are still in the process of deciding your future career, and I understand the anxiety and uncertainty that comes with that decision. I was no different than you. Ultimately, whichever field you decide on, I urge you to remember that there's a distinction between schooling and learning. Guided learning in school is important but futile without exploration and experimentation. Research, the scientific method, questioning of accepted thinking, probing the unknown, pushes the boundary between the known and unknown, not only for you, but for everyone else as well. Engaging in these processes forces you to think critically, self-learn, ask questions that have never been dreamt of. My personal experience with research started with the curiosity of understanding HIV. After pestering a few professors, I had the opportunity to be a summer research intern at the University of Toronto. A few broken beakers later and some more pestering, I've been able to actively pursue research opportunities here at York University, from holding the Undergraduate Student Research Award from the Natural Sciences and Engineering Council of Canada for 2 consecutive summers, to investigating my own project for my honors thesis. You're all potentially the next generation of groundbreaking scientists. With that in mind, I'm excited to present to you 2 inspiring biomedical researchers who will give you new insights into what it is like to pursue science and research today. I'm now honored to introduce our first speaker, a 2014 Canada Gairdner Whiteman Award laureate, Dr. Salim Yusuf. Dr. Yusuf is an internationally renowned cardiologist and epidemiologist. With over 900 articles published in refereed journals, he is the leading North American authority in his field. And in 2011, he was the second most cited researcher in the world. His work in large-scale clinical trials that span dozens of countries across all continents and involve tens of thousands of people have led to improvements in prevention and treatment of cardiovascular disease. Dr. Yusuf currently leads the largest ever study illustrating the role of societal changes such as urbanization, changes in diet, on behaviors and risk factors of cardiovascular disease. Born in southwestern India, Dr. Yusuf obtained his medical degree from Bangalore University in 1976, and later completed his doctorate from Oxford on a Rhodes Scholarship. It was at Oxford where Dr. Yusuf became involved with studying cardiovascular disease. Discontent with the ambiguity of standard clinical trials to show the efficacy of treatments, he, along with others, designed large, simple trials and meta-analyses. In 1984, Dr. Yusuf moved to the National Institutes of Health in the United States and continued to refine this approach and his work in studying treatment and prevention of cardiovascular disease. In 1992, he moved to McMaster University, where he is currently a professor of medicine at the Michael G. DeGroote School of Medicine. He is the executive director of the Population Health Research Institute and vice president of research for Hamilton Health Services. He also serves as the president of the World Heart Federation and holds the Heart and Stroke Foundation of Ontario Research Chair. He's the recipient of 35 national and international research accolades, the Officer of the Order of Canada, and has been inducted into the Royal Society of Canada and the Canadian Medical Hall of Fame. Dr. Yusuf will now talk to you about careers, academia, research, and his journey in science focused through his renowned research. The title of his talk is A 35-Year Adventure in Preventing Cardiovascular Diseases. So please join me in welcoming Dr. Salim Yusuf.
Good morning, everybody. Morning, that's good. I remember going to class and, we had to stand up and say, "Good morning, teacher." I guess you don't do these things again. So thank you, Vakar, for those wonderful words. If my father were here, he'd really be proud. And if my mother were here, she'd even believe it. Now, most of you want a career in science, and I must say, I never thought I'd get into science, let alone medicine. I was born into a middle-class family. My father was the youngest of 8 children, and the first one from his family that ever went to university. My mother was the only child, and being from a Muslim Indian background in rural India, hardly any woman went to university. She did too. So I was fortunate in having parents who cared about education. Now, education, the first thing I remember were 2 things in my childhood. My father would buy me a storybook, but I had to read 1 chapter every day and summarize it in 1 page in my own handwriting and in my own language. He would check my handwriting, he would check my grammar, he would check my expression, and that was 2 of the 3 R's. And I think I did this for many years. When I finished a book, he'd get me another book, and it went on. My mother would sort of rehearse with me every morning, waiting for the school bus, the multiplication tables. Now, how many of you here have had your parents involved in one way or another with your school homework, your reading? Just raise your hands. I would expect to see 100% or practically all. And that's a story that tells you learning starts at home, and it starts with your family. I was fortunate because as I grew up, I wasn't that good a student. I was okay, maybe in the top 5 in the class. Whenever the report cards came, and I got reasonable marks and was very proud of it, my father would ask me, "But what about the rest of the marks?" Now, if you're immigrant children, you probably can identify with that. Yes, I can see that going. But I can tell you, I had a great deal of fun in school. I played a lot of sport, enjoyed it a great deal. My brother, who was a little younger than me, was very good. And because I was a little older and therefore a little bigger then, now he's bigger than me, not hard to do that. But he turned out eventually to be better than me. But we always were together and we did things together. Now, one of the most memorable things that happened in high school, impactful thing that happened in high school, was when I was in 9th grade. That's when in India you decide whether you go into medicine, engineering, arts. And the first biology monthly exam, I got a bare pass. I was furious. I went to my school biology school teacher and said, I should have got better marks. I think I did better. And he turned to me and said, yes, you should have gotten better, but you were lazy and you were coasting. That immediately made me think I had to work harder and show him what I could do. So I went to the university and borrowed university textbooks, studied from it, and the next month I did far better and nearly got a perfect in biology, which was virtually unheard. But that also taught me something, that in most of us there's a lot of hidden capacity only if we discovered it and worked hard. So I really thank both my parents and my biology teacher for the very fundamental lessons that I started life with. Now, my father wanted me to go into medicine, and that's why I went into medicine. It wasn't because I wanted to save the world or discover wonderful and weird and fantastic things. If you're an immigrant child, how many of you have been told by your parents what career to choose? Don't hesitate, raise your hands. Yep, and you know what? Usually they know you better than you know yourself. So don't think you have to be too Canadian. My kids tell me that they're Canadian and we're Indian. Now, how many of you told that to your parents? Raise your hands. Yeah. So, but remember, they know you well. They always want to do the best for you. And although it may sometimes grate, listen to it, even if it's not immediate, even if you reject it immediately. Anyways, I applied to medical school when I finished high school, and the first 4 medical schools I applied to didn't even give me an interview. I got a very nice, polite letter saying, "Dear Mr. Yusuf, thank you for your interest," blah, blah, blah. I actually joined a local college to study sciences, and at that time, I then suddenly got an interview from the 5th medical school, which is where I eventually went to, and I thought I had no chance of getting in. In medical school, the first year was easy because it was really a transition year. So you were really repeating what we did in high school, and that was a year of great fun. You left home, you had your own bank account, nobody checked when you got up, nobody checked when you got home, and it was great. Freedom was great. But it was good to have that year. You're all going to get that year when you leave home and go to university. The key thing is how you use that year. Have fun, but also start to build your steps further. Then second year is when you started to do real medicine in India. And we had anatomy, physiology, and biochemistry. Physiology and biochemistry was reasonable, and most of the class passed its first exam. But anatomy, every single one of us failed. Every single one of us failed, because the amount of material that we had to absorb and regurgitate was just about 10 times more than what we were prepared for. And that's what's going to happen to you when you go from school to university. You suddenly have to learn much faster, absorb things much quicker, digest it and integrate it. So be prepared for that. If high school is tough, it's going to be tougher in university, but you'll manage. So we all failed. The next weekly test, these were weekly tests. Again, everybody failed. The third exam, 1 person passed. The 4th exam, 2 people passed, and they were both girls. I can tell you girls work harder than boys, generally. And the 5th exam, about 10 of us passed, including me. And around that time, my father wrote to me and said, are you keeping up with the rest of the class? That was an easy one to answer. When everybody failed, I could say, yes, I'm pretty average. So anyway, we worked very hard, and as it turned out, I did manage to get the highest marks in the first semester exams, which was a big surprise to me. But that once you get there, you never want to give up. And you try hard and go through the rest of medical school working just as hard. Happened to me in high school, happened to me at university, but I had to lift my game up. We had great teachers. They motivated us because they wanted to bring the best out of us. They were tough as nails. It's not like today where teachers are meant to be friendly with students and friends with them. Our teachers were pretty distant, but they were still inspirational figures. And they had, they were wonderful teachers. When I finished medical school, a friend of mine, whom I admired and who was about several years my senior, had received the Rhodes Scholarship. So he wrote to me and said, "You got to apply for the Rhodes Scholarship." And I said, "You must be joking." So he said, no, do apply. So I put my application together, and then I was extremely fortunate. Another person who was the brother of one of my faculty and who was a Rhodes Scholar from India, was visiting the medical school, and she introduced me to him, and he went over my application and gave me a number of tips, especially my statements, and it transformed it. Now that was crucial help, and I got invited to the interview. Now, in those days in India, there were just 2 Rhodes Scholarships for all of India, and the Rhodes Scholarship isn't in any field, so about once in 5 years, somebody from medicine would get it. When I got to the Rhodes Scholarship interview, there were 12 people. They were all fantastic. They were handsome, they were tall, they were, that's not hard compared to me, but they were just brilliant people, and some had actually represented India in the Olympics. So there were 2 of us, one a sort of thin, scraggly-looking fellow from Pune, who was in English literature, and I in medicine. We looked at each other and said, we have no chance of getting this. So we had a good time. There was a reception. We both had a beer. Having a beer in India in those days for a young person with senior faculty is a no thing. But we had a beer, and we ate food. Free food as a student, getting free food is pretty good. So terrific food. And we went around chatting with the interview committee. And the interview committee, and I was so impressed by what my other interviewees were talking about, how in a certain film in India, the heroine had half her face in the light and half her face in the dark, and that reflected her state of mind and that she was confused as what to do. And I said, oh my God, I saw that film, but I can't remember that scene. So all these conversations were happening, and I had some very nice conversations with a couple of the committee members of my work in the villages of India, where I was working at that time as an intern, doing my rural posting, and we were working on trying to deal with an epidemic of waterborne outbreaks. And I looked at the books, and the best way was to chlorinate the wells. So we needed to chlorinate the wells. Now I could see in the clinic there were 6 sacks of bleaching powder, which I had learned from my medical school books was the thing to use. So with the help of some high school students like you, with whom I'd play volleyball every evening, we said, let's check the quality of the water in the well. So we went to about the 12 wells in the village, took the water up, put it into big containers, transported it to the public health laboratory in the city, and got it analyzed. 11 out of 12 wells came back severely contaminated. Their toilets were right next to the wells. The cow sheds were right next to the wells. So we couldn't do anything about where their toilets were or their cow sheds were, but we thought, let's purify the water by putting bleaching powder, but we had to measure it. So we took a stone, a rope, put it down, and where the rope sort of stopped being wet was the depth of the well. Fair enough? And then we got the diameter, the simple arithmetic, and we calculated the volume of water, and the degree of contamination in the well from the public health lab told us what is called chlorine demand. We calculated it. And so we went to the village headman and said, we think you've got this problem, lots of your children are having diarrhea, we've got to do something about it. So they said, what would you like to do? We said, we'd like to put some bleaching powder into this. And they said, you can chlorinate it, but you got to do it so that the women never know about it, and you do it in the middle of the night. So we, so around 7 in the evening, we started to measure the amount of chlorine needed in each well. After 9, we got into bicycles with sacks of chlorine, bleaching powder, and went around this. So the 8 or 10 of us did. We chlorinated the wells, and when we finished chlorinating, it was around 5 in the morning. And imagine a scene in India. The sun is just rising, it's still mostly dark, the cocks are crowing, and the women are coming down to draw the first amount of water from the well. So they came down and they said, oh my God, you've spoiled our water, we can't drink this. So in a moment of bravado, I put the bucket down into the well, and we drew up the water, and I just wanted to show them, it was very drinkable. I put it down into my mouth and spat it out instantly. It was the same bucket that had the bleaching powder. So that set my efforts at public health bad, because I hadn't worked through all the possible problems that I would encounter in my experiment. Anyway, we put that into, but we retested the water. All 12 wells were clean, and the rates of diarrheal diseases plummeted, and that was my first research paper, and that was what I discussed at the Rhodes Committee, and that's what got me to Oxford. Now, when I went to Oxford, I was fortunate to work with some wonderful people. But when I went there, I had barely used a knife and fork. In India, as most of you know, we use our fingers to eat. So when I went into hall, there would be this plate and several knives, several forks, several spoons, and I was always half a course behind. I would look at the chaps opposite and then sort of follow which cutlery they would. Now, that's just an example of culture shock, not knowing. I was also impressed by the way the Brits spoke. Have you ever noticed how they speak on BBC? It's almost like they're spitting words out. And so it took me a while to understand them, but then soon I learned how to spit as well. So started to become a little English, then a lot more English. But what is amazing was the smugness of people in England. I met somebody and one of these other students in the middle common room at Oxford and asked him, what are you doing? We chatted, and he said, I'm doing, my thesis is on 18th century Indian history. I said, wow, you must really have been to India many times. He said, no, I get everything I need here from the Bodleian Library. I don't have to go to India. That just showed you how much Britain was the center of the world for a certain period of time, and they felt they didn't need to go somewhere else to learn. Anyway, I had a great time in Oxford the first year, but I was also frightened. I had never done research. I had no intention of going into research, honestly. But when you're in darkest, deepest India and you get a scholarship to go to Oxford, you'd say anything to go to Oxford. I'm sure you will do the same thing to get into medical school or whatever it is. But at least there should be half a bit of truth when you do say those things at the interview. Anyway, I'd go to my supervisor every week and say, am I doing okay? And he'd pat me on my back and say, fine. I then got a scholarship to go to Sweden, to Göteborg, where I was last week, and received a honorary doctorate, and they were very generous. I learned from them that the work that I was doing in Oxford was really hopelessly bad, And so they shared their ideas, they shared their protocols, and then they said, when you go back, you've got to talk to this chap called Richard Peto, who incidentally received the Gairdner Prize about 10 years back and might one day get the Nobel Prize. Anyway, Richard was a young chap then, and so when I went back, I called him up and said, "Sir, please can I come and see you?" Now, if you are from an immigrant background, you know the word sir is very important, so I did that. Now, he said, "No, go away, I don't have time for you." So I said, "Please, I know my DPhil is going, DPhil is Oxfordese for PhD, is going badly. I need, and I've been in Sweden, and so Hans Roydell told me to meet you." Said, "Oh yeah, I know Hans, come and see me, but I'll give you 15 minutes. So I went in to see him. And I went into this room, and the piles of paper all over the floor. And it was almost like a hurdle race, and you go in and you sit down, and he said, there was this young looking guy, early 30s, in jeans, torn shirt. He said, "Do you want coffee?" And I said, "Sure." And then when I looked at the mug, it was very dirty. I wish I had told him, "No, I don't want coffee." But anyway, he gave me coffee, and the 15 minutes became 3 hours that day. And then to this day, 35 years later, we still collaborate. So another lesson I learned was, when you have problems, go to the experts, bang on their door, be persistent, even harass them, pester them, as I think Walker said, and persuade them that you're serious and they'll help you. Now, we then I then realized, based on Richard's comments, that what we were doing was hopelessly inadequate. We needed to do much bigger studies, and to do much bigger studies, we had to collaborate with people around the world. So this required persuading other people to work with you and share in your vision. And this wasn't easy when you're in your mid-20s, but with the help of my professors and Richard Peter, we did. And that was then the birth of these large multi-center studies that actually now is standard around the world, and has led to the discovery of many, many treatments and preventive strategies that have saved lives. Now, all along, I was afraid of failure. It wasn't that I was dreaming of success. I just wanted to get my DPhil and not be a failure and then go back into clinical medicine. But the more I got into research, the more I got hooked. It was like an addiction. I did go back into clinical work, trained, which is the equivalent of doing my residency program, got my membership, which is like getting the fellowship here, did my cardiology training, but I couldn't wait to get back into research. Nights and weekends, I'd go back to the lab, work on things. And then the NIH offered me a job, which was totally unexpected, out of the blue. They were looking for somebody who had clinical trials expertise, but was also a clinician, and the job suited me, and that gave me the opportunity of developing the first major program in the world in heart failure. And it was a fantastic time at the NIH. People at the NIH sort of thought that they could solve all the problems of the world. They had the confidence or the arrogance, depending on the way you put it, that no problem was beyond them. And we had endless hours of discussion and arguments and over lunch. And the most important thing in research is to think, to discuss. And if you read Watson and Crick's biography, you will say, they said, "We didn't do much work, but we discussed and thought a lot." You will find medicine today is doing, and research is much more thinking, although more and more people are looking at research as, how many papers do you have, where did you publish? Those happen, but your primary goal needs to be to reflect on everything. So I'd learned that I had to work hard, I'd learned I had to collaborate, I'd learned that I had to be humble and seek help, I'd learned that other people would be generous if you only asked, I'd learned that you had to dream big ideas. Then I came to McMaster, and we developed a fairly significant program. And I then moved into my current phase, which is working on global health. As you know, I'm from India. I wanted to do something that had an impact on developing countries. And most of you may not know, but 80% of the cardiovascular disease deaths occurs in poor, low and middle income countries, yet practically no research had been done there. So over time, we've been doing studies in 50 countries, and the way we did it is not because we had money, we had an idea, we kept it simple, and we sold the idea to other people, and they became our friends, and friends became our collaborators. Throughout life, no matter what you do, research, medicine, anything else, architecture, business, you will always find that you need to first make friends before you make collaborators. So that is another lesson that I've learned, because they will help you that goes way beyond money. Now, my role today is mentoring people, working with people, and there's no more happier moment than I have than my weekly meeting with my close research team. I head an institute with about 320 people, but there's about 20 people in my close research team, and we meet once a week. We discuss ideas, we discuss their papers, we discuss problems, and everybody is helping each other, and I learn from them. I have no formal training in statistics or epidemiology, so I'm learning from my fellows and PhD students. And one of the things when I take any PhD student on, in addition to assessing the usual stuff, I also tell them, what will you be able to teach me? Because I believe every student should be able to teach their mentor. Otherwise, so that is my lifelong learning as I go along. Now, as you grow up, and I'm still growing up, hopefully not sideways, your goals will change, and that is completely okay. I know when I was very young, I wanted to be a steam engine. Not a steam engine driver, but a steam engine. Then later I wanted to be a fireman. Then later on, sort of, maybe a doctor. But then I thought I'd lost my way, and especially when I saw the movie Star Wars, which all of you might have seen, and I said, gosh, the villains in life have the greatest fun. So I wanted to be Darth Vader. But then when I had a bit of sense drummed into me, I said, well, maybe Obi-Wan Kenobi might be better. But then more recently, I thought maybe I should be Yoda. Small, shrunken, shriveled, but with wisdom. And that's where I'm headed to right now. So thank you.
So, thank you very much. We have time for a few questions before our next speaker. There are microphones that will be brought around. For those of you in the balcony, I'm not sure we have any microphones there, so you'll have to come down because we can't hear from up there. The other thing, I'll do my best, but I'm not an actor, and the lights on this stage are abominable. I can't see people out there, and I'm sure Dr. Yusuf is having the same problem. So
yeah,
just
I
put
can
your
see you
hand
now.
up,
Yeah,
and we'll get a microphone to you as quickly as we can. So there's a question right here.
or just speak up. Yeah.
Hi. Through your whole path of education, did you ever find yourself doubting yourself when maybe you weren't top of the class or whatever? Do you know what I mean?
Oh, I doubted myself right through life till about a decade or so ago. I became confident in my 50s. Till then, I wasn't sure what the hell I was doing. In fact, to be honest, our big study that Walker told us about, till the first results came through, which took 10 years, I wondered whether it was my biggest folly. So doubt is a good thing, but don't let doubt stop you. And also remember you're going to have obstacles, and you need to be prepared to find alternative solutions. So yes, I have continually doubted myself. And I think doubt is good, but don't let doubt paralyze you.
Okay, other
Right, just stand up and
back.
yell. Yeah.
Yeah, there's a microphone coming.
Hi, I was actually fascinated by the fact that you said that it's impoverished nations where cardiovascular diseases are such a huge killer. And I was wondering if you could maybe discuss what you think some of the reasons are. I always thought it was the excesses of the Western world that lead to high cardiovascular disease rates.
I think there's no doubt that the excesses of the Western world do increase your risk factors. But smoking is a lot in the developing countries. Poor diets are very high, and although obesity is less. But diabetes rates, paradoxically, are 4 times higher in poorer countries, and we don't understand that. The other thing is, once somebody gets subclinical disease, it's picked up earlier in Western countries, and we do something about it. These countries, they're not picked up. And once somebody gets a heart attack or stroke, the mortality is 600% higher in the poorer countries. So actually, although overall risk factors are lower, smoking is higher, and diets are poor in these, and the progression from a healthy person to unhealthy but not clinical disease is also more. And like blood pressure control is less, cholesterol control is less, and quitting smoking is less. And then preventive strategies and treatment strategies are weaker. And that's what we're now working on and trying to rectify it. And there are barriers. Even simple, cheap drugs are unaffordable. Generic drugs are unaffordable in 40% to 50% of the people in poor countries. I mean, that just astounded me. We actually have a paper that's either come out today or coming out tomorrow in the Lancet showing that. It surprised us completely. So we need, until we get universal health care in these countries, which means access to health care is better, we're going to have big problems. And you know, the poor countries, 95% of the healthcare costs are paid by patients from their own pocket. And often they have to borrow money, sell land, sell property for when they have catastrophic illnesses. We really are fortunate in Canada. We can complain about our healthcare system, but the fact that when you're unwell, you don't have to worry about how you pay for it or getting good quality care is just fantastic.
Okay, yeah, that's fine.
Hi, you said that when you were in medical school, you worked with high school students, like they helped you to chlorinate the water, and
Right.
you did like that kind of research project together. So do you know how us high school students can go about finding opportunities for ourselves so that we can kind of like find a path where we want to go in medicine and get some early experience?
Yeah, the first thing I want to tell you is I didn't think what I was doing with the high school students was research. And before we even approached them to do something, you know, every evening the volleyball court was behind where the doctors lived. And so I got to become friends with many of the kids. So when we discovered this problem and we had the idea of doing something, I sort of said, how about doing this? So it wasn't your classic research protocol where you had a protocol and you went stepwise. It just happened. It was a natural thing to do. There are many ways you can do it. One is to find a researcher somewhere, you know, social sciences, medical sciences, biological sciences, anything, you know, chemistry, physics, anything. And you could say, look, I'm interested in. The other way is you can start to look around you and see what are the problems in your community. And if you, and then you say, are any of those problems small enough for you to start to tackle it? Because you can't really tackle something big. And then you start to ask the question, and then you take that question to somebody who may have the skills and persuade them that you're really serious, because remember, supervising a student is actually tough work. To supervise them well, you have to give them a lot of time. And so I, for instance, reject 95% of the people who write to me simply because supervising a student takes me 4 to 6 hours a week. And I don't take more than 3 students at a time. So I think what I would say is, if there is a burning question, if you go around saying, I want to do research, it's not going to happen. okay? So you want to be a little more specific. It could be, on reflection, not the brightest question, but it is your question. And I would start with that, then seek the help of somebody to answer it. And depending on your interest, it may be a biological one, it may be a sociological question. They're all good questions. So there isn't a single way, but if you truly want to do research, it has to come from inside.
Okay, I think there was one question sort of at the back. Did somebody have? Yeah, right there. And this will have to be the last question, but just wait for the microphone.
What would your greatest piece of advice be for an aspiring doctor or scientist?
The first is ask yourself whether you really want to do it, because it is a tough, tough grind. There are easier ways of making a living. So ask yourself that question first. Being a doctor and being a scientist is fantastically exciting. I can't tell you how wonderful my work is. I don't think of it as work. It's fun. But the thing is, you really will need to want to do it. And especially, you see, being a doctor, the moment you get into if you manage to get high enough grades and do well in an interview and get into a Canadian school, and you work hard, your life is ahead of you. It's all set for you. Because then it's almost like a tunnel. You make it through to the other end. But science is different. If you don't ask the question, nobody is going to tell you what the question is. If you don't have the initiative to design your protocol, nobody's going to do it for you. If you don't go out and do your experiments, nobody's on your back to do it. Nobody gives you money to do it. You have to raise the money. Science is one order of magnitude tougher than being in medicine. And you can be a good scientist without doing medicine, and you can be a good doctor without doing research. So, but you can be very good doing both. So they're not mutually exclusive. But the bottom line is that you really, really want to do it. And if you really, really want to do it, you're going to spend whatever time it takes, accept numerous disappointments, because me and Michael definitely had more grants rejected than funded. I mean, at least that's my case. Maybe you've got a better track record, Michael. So you need to be able to pick be disappointed one day, but pick yourself up the next day and then get on with it. So, good luck. I hope many of you will go into science. And there are different ways of going into science. I mean, you don't have to, it's like a pyramid. Not everybody gets a Nobel Prize. And in fact, somebody who says, I want to become a neurophysiologist of the hippocampus at the age of 2 really doesn't know what they're talking about. So just go step by step, dream the next way, and go along. So good luck to all of you, and thank you.
Okay. So thank you very much. We'll move on now. And I'll ask Walker to introduce our next speaker. So thank you very much.
Thank you, Dr. Yusuf, for your inspiring words. I'm now pleased to introduce our second speaker, Dr. Michael Salter. Dr. Salter is an accomplished scientist and is currently a member of the Gairdner Medical Review Panel. With publications in elite journals such as Nature, Cell, Science, Nature Medicine, and Neuron, he is a pioneer for furthering our understanding of molecular and cellular mechanisms of neuroplasticity. His current work focuses on synaptic physiology and uncovering the fundamental principles of cell-to-cell signaling in the central nervous system, and their effects in signal transmission, pain, and plasticity when alterations or aberrations arise, such as in disease or pathology. Dr. Salter obtained his medical degree from the University of Western Ontario in 1982 before receiving his doctorate in physiology from McGill University in 1987. Following postdoctoral training at Toronto Western and Mount Sinai hospitals, he joined the Sick Kids Research Institute and has been a member there since 1990, and now serves as the Chief of Research at the Hospital Research Institute. He has held progressive leadership positions that include senior scientist, the head of program in neurosciences and mental health, and the associate chief in science strategy and commercialization. Since 1999, Dr. Salter has been a professor in the Department of Physiology, the Institute of Medical Sciences, and the Faculty of Dentistry at the University of Toronto. Also from 1999 to 2009, Dr. Salter was the founding director of the University of Toronto Centre for the Study of Pain. His scientific expertise and leadership is highly sought after. Dr. Salter is a founding scientist of 2 startup biotechnology companies, both involved in developing novel drug treatments. Among his many accolades, Dr. Salter is a fellow of the Royal Society of Canada and holds a Canada Research Chair Tier 1 in Neuroplasticity and Pain, the Anna Max Tannenbaum Chair in Molecular Medicine at Sick Kids, and the Northbridge Chair in Pediatric Research. He's the recipient of the E.B. Eastburn Award, the John Charles Polanyi Prize in Physiology or Medicine, the Early Career Investigator Award, and the Distinguished Career Investigator Award of the Canadian Pain Society, and was an international research scholar of the Howard Hughes Medical Institute. Dr. Salter will now speak to you about careers, academia, research, and his journey in science focused through his renowned research. The title of his talk is Chasing a Dream in Science. So please join me in welcoming Dr. Michael Salter.
Thanks very much. Well, thank you, Walker, for the very nice introduction. So I must say, note to self, never follow Salim Yusuf. Okay, let's see if we can get this thing up and running. So I thought I'd take a slightly different tact. And first, I should remember, it's Back to the Future Day, so happy Back to the Future Day to all of you. You guys know what that means? Okay, good. I thought it was just me, maybe Yusuf also. So I thought I'd start off by talking a little bit about where I work now. So this is where I work. This is the Research Institute at the Hospital for Sick Children, a building that we built and started to occupy a couple of years ago. And if you talk to anyone from Sick Kids, they will tell you what a great, the researchers especially, they'll tell you what a great building we have, right? You can see it's a beautiful new building. We have wonderful labs, lots of space to make discoveries and do all kinds of interesting and provocative things. But really for me, as Chief of Research, what's the most important part is not the building, but what's inside, which is the people. It's the people that come up with the discoveries. Every day I get the pleasure of walking into this building with about 250 faculty members and over 2,100 scientists, students, technicians, research fellows, all working to promote human health and to discover things. Every day in this building, somewhere in this building, somebody has thought an idea or thought a thought that's never been contemplated in the history of humanity ever before. It's interesting to think about this, right? This is the kind of thing that drives one over one's career. You heard that from Salim. Thinking about great ideas that no one else has ever thought of. This is the kind of contribution that one can make. This is what drives us all that stay in science for many years. It's the people and it's the great ideas that we try to follow over time. So for me, even though Salim, you said you couldn't really trust someone who wanted to be a neurophysiologist at the age of 2, my interest in the brain started around 4th or 5th grade, maybe, thinking about this great uncharted part of the human body, this most complex structure that we know about in the universe, the human brain. This is actually a picture from a special imaging technology that we use at Sick Kids of one of my kids' brains. So this was when he was, I think, about 16 years old. My wife thought we should put him in a research study because she wasn't sure if he had a brain, being a 16-year-old boy. So this was proof, formal proof that he does have a brain. And I use this just as a segue to show you a little inspirational video, or a video that I think is inspirational about what we think and what we know about the brain. So I'll just put this on. So this is, I think you can see, an important organ, something that we know very little about, and really 95% of what we know about the brain we've learned in the last 10 years. So it's an area ripe for young students, or even old ones like me and Salim, to continue on, to think about. So this idea about the unknown, having great curiosity about something that really drives you, has really been the underpinning of things that I've done over the last 35 or 40 years in all the research that we've been doing. Thinking about how is it that we use this brain of ours to dream great thoughts, to do all the things you saw in the video, all those wonderful things that we do as human beings. But one question that keeps driving me is, are we really ever going to understand how the human brain works? And that's a continuing question. Just 2 days ago, I left the Society for Neuroscience meeting in Chicago. There were 35,000 scientists there. That's about roughly maybe a quarter of all the scientists in the world that are studying brain function. So can you imagine 35 brain geeks all standing around thinking about their ideas about how the brain works? It was really exciting and provocative and interesting. So this is a field that continues to grow and develop, and one that really has, for me personally, driven me for the last, all my career. So we know a lot about the brain, and I'm going to tell you a little bit about the kinds of things we know. So this is another example of a brain imaging picture. Happens not to be from SickKids, but from somewhere else. So we've learned quite a lot, even though we don't really know that much. We think we've learned a lot about long tracks, about how the brain works, and how various parts of your brain are connected together to allow you to do things like move around and talk and think and run. But when it really comes down to it, it comes down to cells in your brain working, and cells in your brain talking to other. To paraphrase Carl Sagan, you guys know who Carl Sagan was? You don't know who that, oh, good, one. Okay, there's billions and billions of neurons in your brain, and they make trillions and trillions of connections. But this is a relatively new idea in the history of human thought. It was really an idea from this fellow. Anybody know who this is? No? Ron? Don Santiago Ramon y Cajal, okay? Started studying brain sections and realized that the brain is full of individual cells, nerve cells, in his lab back in the late 1890s, right? And I put this picture of Cajal up here because we think a lot about what we know now comes from what we learned in the last decade. I told you that. But the fundamental underpinnings about the way we think about the brain all came from Cajal. Cajal scholars say everything we know about the brain came from Cajal, right? Everything we know came from the Greeks, right? So this is Cajal. This is where the father of neuroscience, the person that taught us really how to understand and how to think about the brain. And I told you, it's really connectivity within the brain that really is what we're interested in my group and in many groups around the world. Connectivity from these long tracks that connect various parts of your brain together to the little microcircuits that connect individual nerve cells to one another and allow us to process information and to actually do functions. And so we know quite a bit about this, and this is a video that one of my students put together that I'll show you next that really can give you a sense about some of the very deep ideas we have about how nerve cells in the brain connect to one another. So let me just see if I can get that going. Okay, this is about a phenomena we think underlies learning and
long-term potentiation, or LTP, refers to a form of synaptic plasticity that occurs following periods of intense synaptic activity. On the left, we see synaptic transmission at normal levels of stimulation. As the frequency of stimulation increases, however, transmission at the synapse becomes more efficient, resulting in the amplification of impulses across the synapse, as seen on the right. But how does this happen? During baseline synaptic activity, glutamate is released into the synaptic cleft. Here it binds to glutamate receptors, resulting in the depolarization of the postsynaptic cell, and thus impulses are transmitted across the synapse. These receptors are embedded in a highly organized structure known as the postsynaptic density, or PSD. The PSD is a dense lattice composed of neurotransmitter receptors, signaling molecules, and the cytoskeleton. In the postsynaptic membrane, two types of ligand-gated glutamate receptors can be found. AMPA receptors are responsible for the majority of the postsynaptic response at low levels of activity. Current flow through NMDA receptors, however, is suppressed by an extracellular magnesium block. As activity intensifies, however, increasing intracellular depolarization becomes sufficient to repel the magnesium blockage, allowing current flow through receptor pores. NMDA receptor current is further boosted by the tyrosine kinase SRC, which is anchored at the receptor by the protein MD2. SRC then phosphorylates the receptor, further amplifying its activity. Importantly, however, NMDA channels are permeable to calcium. It is this calcium influx that is the critical trigger for LTP. Calcium exerts much of its effects through the kinase CAMK2. Upon activation, the enzyme translocates to the PSD, where it directly phosphorylates AMPA receptors, further enhancing their activity. The second action of CAMK2 is to dramatically increase the number of AMPA receptors on the cell surface. Vesicles containing pre-assembled AMPA receptors form an intracellular pool at the base of the dendritic spine. Exocytosis of these vesicles can be induced to deliver their receptors onto the cell surface. Surprisingly, however, exocytosis does not occur directly into the synaptic cleft, but rather at extrasynaptic sites. Through unknown mechanisms, lateral diffusion of these AMPA receptors brings them towards the synaptic cleft, where they will be temporarily immobilized and ready for further glutamate release.
This is my favorite part.
The result of these molecular events is either an increased activity of individual AMPA receptors or increased number of AMPA receptors on the cell surface. This increases the magnitude of the postsynaptic response to neurotransmitter release, resulting in the amplification of inputs across the synapse.
So you learned Neurobiology 101 in about 5 minutes there. And the reason I showed you that wasn't just to try to tell you how important neurobiology is, but also to make the point that although scientists think most of what we do is work in a lab or come up with great ideas and do great experiments, some of the most important thing we do is communicate with other people. If you have an idea and it stays in your head or stays in your own lab, it's not finished. It's not out there for other people in the community, in the world, to take advantage of, to build upon, to come up with their own great ideas. So communication, I used to tell this when I would go into my son's grade 4, grade 5 classes. They would say, what's the most important thing to do in science? And I said, come up with great ideas and communicate effectively, right? So many of you will think, you want to go into science because you want to hide away from other people, right? People, I told you, are the most important thing, and being able to communicate effectively is equally as important. And that's certainly one thing, a hard lesson that I think many of us learn, because many of us do go into science because we want to get away from other people. Okay, so I grew up in London, Ontario, not too far from here, right? Also in a relatively middle-class family. My dad was a high school teacher. He was actually a high school phys ed teacher. And so during high school and during elementary school, back in the 1960s, the space race was on, right? So this engaged many of us greatly, to the quest to get the race to get to the moon. It was a great geopolitical battle between the United States and the USSR at the time. And of course, you know that in that race, the US won, putting the first person on the moon. On July 21st, 1969, Neil Armstrong stepped on the moon. I remember sitting in my living room with my parents and my 2 brothers, and we watched as Neil Armstrong stepped onto the moon. So this was a great advance for technology, a great advance for science, and really, for many of us, I think, highly inspirational. For me personally, very inspirational to think of the kinds of things we could do. We could actually put somebody from our planet on another heavenly body somewhere out there. So this was a great motivator, and Neil Armstrong certainly was one of my early heroes. At one point, I did want to become an astronaut, but then my myopia set in, and that kind of put that to rest. So. But also in those times, as part of this geopolitical battle between the West and the Soviets, we had other heroes. Anybody know who this guy is? Yes? Yeah, good try. This is Paul Henderson, someone you should know, scored the winning goal for Canada in the Canada-Soviet series. Who's this guy? Bobby Orr, of course, everyone knows Bobby Orr. But Paul Henderson, right? I have a Paul Henderson jersey in my office. So, sports heroes, heroes in sports, heroes in life for me were very important. I think for many of you, they're very important. You think of sports heroes, these were some of my sports heroes. Today we have sports heroes like this, being in Canada. We like to pride ourselves, I'm gonna fall apart here. Pride ourselves, especially on hockey. But lately in Toronto, there's something else we can pride ourselves on. You already know who this guy is. I was at the baseball game last night. Man, what a disaster. Unless you're a Kansas City fan. It was really, really very sad. But they'll come back today and they'll win the rest of the series. So being an optimist is critically important in life, in sports, and in science. Salim told you, and I'll tell you again, dealing with failure, learning how to deal with failure is one of the most important things that we can all learn, right? As I said, the Blue Jays are going to come back. I know that for sure. We all have papers rejected. We have experiments that don't work. Actually, many of the most interesting, salient, and provocative discoveries that we make are failed experiments, where you think one thing's going to happen, and something else completely different happens. And what you have to do is be prepared to see that. Some of those things that we do, certainly in my lab, many of our most important discoveries have been based on experiments that we thought were going to go one way and turned out a completely different way. But then one needs to have the perspective of saying, okay, maybe we were wrong about that, and maybe there's a different idea and a different way of thinking. And challenging and being different in the way one thinks not only is good in sports, but also it's good in science. So who knows who this guy is? Well, I put his name up there already. So you heard already from Ron Perlman about Art McDonald winning the Nobel Prize just a couple, or having it awarded just a couple weeks ago, right? So here's a person from relatively humble beginnings, like many of us, who's gone on to be one of the top scientists ever in Canada. And so I think this is something that's inspirational, aspirational for us as well, thinking big and following what you can do. And that kind of motivation is what really has driven me on since I told you I started thinking about the brain and about the space race many, many years ago. Okay, so I come back to this question because we don't have an answer yet to this question, and maybe you guys will be part of the answer to figuring out how the brain works. For me, it's the question has changed now, maybe because I'm getting a little bit older, is are we going to have a sufficiently good answer about how the brain works to be able to do something interesting? You saw in the first video I showed you that hundreds of thousands of people in Canada and around the globe are affected with brain disorders. So can we develop an understanding of how the brain works that's sufficient to allow us to come up with novel therapies? And that's one of the kinds of things that we've done in my lab, and you heard from Walker that we've started a couple biotech companies based on some ideas that we know are imperfect. They're incomplete ideas. We don't know everything, but we know enough to at least try to make a step forward and see if we can improve the human condition that way. For me, one of the things in this universe of different areas of brain science has been something that I can relate to one specific lecture in medical school by one of my physiology professors who started talking about pain and started talking about this crazy idea, at least crazy at the time, idea that the brain can essentially control how you perceive pain. And this got me thinking quite deeply about how we use our brain to actually develop and construct our entire universe. So everything that you see around you, everything that you hear really is a construct of your brain. You live in the world constructed by your brain. And so things that happen to you, your ability to learn and remember and to experience the great joys of life, and also things as devastating as chronic pain, these are all functions of your brain in health and disease. And so we started thinking about that. And for me, this has been something that it's that one lecture in second year medical school that started me thinking, oh, this is something that's really strange. I don't understand it, but maybe that would be something that would be worth following along. And that's, together with this insatiable curiosity about the brain, it's about chronic pain in the brain. And so we've been working about that, and then I motivate the people in my lab with little things like this. So maybe you can't see that from here. It says every day about 1,000 people develop chronic pain. That means every single day we don't discover a cure or a treatment for chronic pain in my lab or in the other labs that are working on this around the world. We doom 1,000 people to suffer from chronic pain. Okay? So that's about one person every single 90 seconds developing chronic pain. So this is one of the kinds of things that we use to try to motivate ourselves to work harder, to work faster, to think more deeply about this problem. So one of the things that we've discovered is a cell called the microglial cell. Anybody heard of microglial cells? No, I can't see. How many people have heard of neurons? Everybody, right. So the microglial cell is the poor cousin of the neuron, right? Very important cell in the brain, as I'm going to tell you in the next couple minutes. But first I'm going to tell you about this guy, Pio del Rio Ortega. Remember I told you about Don Santiago Ramon y Cajal? This was a student, Rio del Ortega was a student of Cajal's. He discovered the microglial cell. Cajal didn't believe him. Cajal didn't believe him so much that he didn't speak to Ortega for almost 50 years, right? But Ortega persisted, developed this field. Cajal was a giant. I told you that really the godfather of, and godfather, I mean godfather, right? Godfather of neuroscience. And yet Ortega persisted and eventually was right. So just because your teachers tell you something, and where are the teachers out there, sorry, doesn't mean that they're right? You have to follow your own dream, your own passion. And without the perseverance of Ortega, we wouldn't have the field of microglia and all the important things we think microglial cells do, even though you've never heard of them until about 2 seconds ago. Okay. Guys in my lab think a lot about them. These are a couple of them hamming it up in front of our some of the equipment that we use. So when you do biology these days, at least the way that we do it in cellular biology, you get to use a lot of cool toys. So you get to play with things. This is the end of a microscope, the other end of the microscope, and I think I put a picture in there. The other end of the microscope is this part, right? And this part of the microscope allows us to see tiny little cells like nerve cells and microglial cells. And that's in large part because of a couple lasers that are here. And this whole bit, this whole thing is worth about 4 Ferraris. So we have to treat this equipment very carefully, but we do get to play with cool stuff a lot when we try to do this. Okay? So we do things like this. I'll show you this little movie. So this is a picture from, it's actually from the spinal cord of a mouse. And in this mouse, we made the microglial cells turn green for your viewing pleasure and enjoyment. And that's the microglial cells. So in between there are the nerve cells, but you can see it's like a lattice work of microglial cells in there. So not only do we get to use cool toys, we get to make cute little videos like this to show you these. But these do have some value. For example, we can study and characterize individual microglial cells. So we've asked the computer here to pick out this one here and spin it around and show us this. So we do this for visualization so that we can communicate our ideas to you guys in a way that you'll remember them. But we can use these images to make highly quantified measurements, and we use those measurements to tell us about how the microglial cells are functioning. So it's not just all fun and games. We actually do some real science in there as well. But one of the most interesting things that's come out about microglial cells is that these cells, sort of unlike neurons, these cells are very active. They're always pushing their processes around, and you can see it here, that one that you can see this one. extending, retracting, extending, retracting. So there's millions and millions of microglial cells in your brain and your spinal cord, and they're all doing this right now. You don't know it, but that's going on in your brain all the time. The microglial cells are sniffing around there trying to figure out what's going on. So without these cool toys and these fancy schmancy, yes, fancy schmancy is a scientific term, these fancy microscopes, we wouldn't be able to learn about these cells and to follow them around and figure out what they're doing. These cells, in normal states, like normal, most of you are probably normal, have normal functions that we're learning about, but they can also be involved in disease, and we're interested in figuring out how they're involved in pain processing. And so that activity that I showed you, one of the things is that the activity I showed you dramatically changes. So you might have seen this part of this microglial cell here. Instead of extending, retracting, this is a big long arm of the microglial cell pulls itself in, and so we think that kind of activity, and I'll show you to you again, is very important for the microglial cells in terms of the way they respond in pain states, right? So these microglial cells now, we have lots of evidence from our lab and dozens of other labs around the world now that microglial cells are very important in chronic pain states, okay? So we have cute little sayings like this: the secret to pain is microglia in the brain. Now you'll never forget that, right? But it's this idea, right? So like Ortega, when we started this work, we kind of fell into it backwards, right? We didn't want to study microglial cells. Microglial cells are the you guys don't know who Rodney Dangerfield is. Rodney Dangerfield of the, they don't get no respect because no one cares about them. Cahal told us these are boring cells. Glia means glue. They're glue, and they're tiny little glue cells, not even big ones. These cells, if you're an immunologist, you think of these cells as part of the immune system. My friend Clifford Wolf at Harvard calls these pus cells in the brain. I mean, how much more denigrating can you get to these cells? But we've come to love and cherish these cells. It's against a tide of people saying, these cells are unimportant. They don't do anything. You guys don't know what you're doing. Right? So that only inspired us to work harder, to fight against people who had contrary ideas, fight in the nice collegial scientific sense, not in the sort of throwing beer cans at the umpire sense, right? And persevere. And I think those are some of the lessons that we've learned over the years, that when people tell you you're wrong, sometimes they're right, that you're wrong, but many times they're not, right? They have their own biases and perceptions, and you have your own way of seeing the world, and your view of the world is just as valid as is theirs, and it's important for you. It's in fact your responsibility to follow your ideas, because as Salim told you, the same thing. If you don't ask the questions, if you don't follow your ideas, nobody else will, and nobody in the next history of the world maybe ever can benefit from your intrinsic brilliance if you keep all this inside yourself. So you really got to let it out. You really got to tell other people. And for many people, like Salim and me, I think we have very similar ideas. We were, very scared about things as we went along. We were not the boisterous people that you see now. We were very timid about these ideas and worried about what other people would think. But one needs to persevere in all these things. So I'm going to come back in the end just about to tell you that this whole area of neurobiology is still one of the great mysteries and one of the great uncharted areas. At SickKids, we have a huge program in neurobiology and mental health going on. We still think about these things a lot. And so really, for me, for you today, really is not only can I be part of this solution, but can you guys be part of discovering how the brain works and putting that to good use. So I'm going to end there and say thank you again to Ron and the committee for this opportunity. I'm happy to answer questions.
Okay, great. So we have time for some questions again. We've got people with microphones there. So if you can put your hands up and we'll do our best to find you. Oh, I'm sure there's some questions. Yeah, go ahead.
The image up on the board, what type of imaging is that?
That's from MEG, magnetoencephalography imaging, yeah. Yep, surface rendered, yeah.
Okay, there's, yeah.
So if you want to perform an experiment, right, on people, how do you find the people to kind of experiment on or to view their brain?
Yeah, so you ask their moms, like my wife volunteered our kids. So it really depends. At SickKids, we use a number of different approaches, and I think it's pretty much the same. We advertise for normal subjects, so this is, you know, to the extent that my son is normal, that's a normal human control brain. But we encourage people, if they're in clinics, if they come and seek out medical help because they have a particular disorder, to think about being involved in research. At SickKids, it's a lot of children. We provide that opportunity to their parents to get involved as research subjects. We encourage children, their parents, and their families to become involved because, I mean, you've seen one side of research here from my lab, and you've heard a very different side from Salim. And in the middle, there are many different kinds of research questions that we ask about individuals interacting with their family and their local communities and globally. So there's many different ways that we can get patients and their families and their communities involved in research.
Down at the front here.
Have you or your fellow researchers come up with like a theory or even part of a theory to how the brain works or how it could have helped you?
So, yes, as I think I was trying to say, we know quite a bit, we think we know quite a bit about how the networks in the brain work, how some of the simple computational processes that the brain uses. Most of these theories are related to neuron-neuron interactions. When I showed you those fancy pictures of the maps and even synapses, you think about neurons and how they interact. So that's kind of the level people are thinking about computation. There's a lot of computation in neuroscience. Actually, a lot of the computational principles in neuroscience have been applied outside neuroscience. So for example, on this device, right, these kinds of devices that allow you to recognize your fingerprint or your face, a lot of those use neural net theory, and much of the neural net theory was developed at University of Toronto. So you can move information, ideas back and forth, right? What's lacking is really a comprehensive theory of everything about how the brain works. Because right now we don't think about integrating neurons and non-neuronal cells, which you know are very important, as well as the microglia. There's another big class of non-neuronal cells called astrocytes that people are just starting to learn about. And some of the really tough problems about the brain involve how neurons and glia work together in both to perform normal functions like talking, sleeping, reading, writing, all those kinds of things, and how the interactions between those cells are disordered in chronic pain and Alzheimer's disease, after a stroke, those kinds of things.
Question back there.
I heard that there was a study going on to see if each neuron had a function. Do you know how far that's gone?
Sorry, where are you? Oh, you're there. Okay. So there's been many ideas about, maybe you're referring to studies about the so-called grandmother cell. Is that the kind of idea where you might have, there's some recording. So you can record the electrical activity of neurons in various parts of the brain. And there's one very famous study about the so-called grandmother cell related to a cell that fired off a lot when the subject was shown a picture, well, it was actually a monkey, was shown a picture of the grandmother, right? So the famous grandmother cell with the idea that each individual cell. The neurons probably work in small groups or clusters, and many of the functions are actually distributed across brain areas. So certainly when we think about pain perception, we know that there's neurons in multiple different areas that are all involved in sort of where does it hurt? How much does it hurt? What do I need to do about it? Does it really bother me? This holistic experience that you have, right, in not just in pain, but when you're looking around and viewing the world, you have this entire holistic view. That requires millions and millions of neurons to work simultaneously.
Okay, question back there.
How close do you think we are to coming up with practical and effective treatments for like neurological diseases?
So I think we all feel we're always this far away, And then something happens and the drug treatment or the other intervention doesn't work, and now we think we're this far away. So for a number of disorders, then it seems that we are on the threshold with some new pharmacological therapies, and some other really interesting kinds of approaches using electrical stimulation. Some of the neurosurgeons really like to stick electrodes into the brain, and these can have very profound effects, for example, in Parkinson's disease, getting people to move again, in depression, and now some of the sort of engineering bias folks are thinking about using brain stimulation of the brain transcranially, so putting large magnets on parts of the brain and stimulating those to do everything from help rehabilitate with stroke to actually making people smarter, kind of the scary part. But so there's so many different people. I told you there's probably about 150, 200,000 people around the world working on these kinds of problems. So there's a lot of good ideas out there now.
Okay, we probably have time for maybe one more question if we have one.
Yeah,
okay.
One there or one there.
Okay, maybe we can do two. There's one in the front.
Yeah, one here.
Okay.
What's the connection between brain and conscience? Do we have an understanding of conscience?
So this is one of the deep questions in neuroscience. It's one that we usually address very late at night in a bar somewhere. So modern neuroscience, the theory is that consciousness is a brain function, right? So that everything that we experience, including consciousness, is a function of various networks of the brain working in various ways. There are a number of alternative theories that have been put around, mind-brain dualism and things like that, but I would say that 99.9% of neuroscientists now think of a holistic view of functions of various parts of the brain. The question is which parts of the brains are involved in meeting which aspects of consciousness. So consciousness isn't really a singular entity, but there's the part of it that's staying awake as part of consciousness, being able to attend to things like I'm looking at you or you're looking at me, that part of consciousness. So different brain regions and subnetworks are involved in that. And so a lot of those details are starting to work out. But I think there's very few dualists around out there in neuroscience anymore.
Okay, there was one question at the front, I think.
Or just right there where she's got the microphone.
Oh, okay.
What kind of ethics regulations do you have, and how do you surpass them?
So we don't surpass the ethics regulations. They're there. That was a plant. What is that? So the ethics regulations, so there's a whole series of regulations for the use of subjects in research, whether they're animals or whether they're humans. And when we're doing research studies, a lot of the studies in my lab and many of us at SickKids work on rats or mice or fruit flies. So there's a whole series of ethical principles that we use in terms of guiding our research and making sure that we want to advance knowledge to improve human health, but we do that in an ethically acceptable way. Ethics extends out to working in humans. So, many scientists think ethics lives over here and science is here. This is kind of a 1970s version of ethics in science. Really, we're thinking about ethical principles in science all the time, whether it's enrolling subjects in to be normal controls or subjects with particular diseases as individuals, or even we have ethics for large community-based trials and ethical principles that guide when you're trying to do an intervention in a community. There's ethical principles that guide that as well. So it's not really, we work within those to balance sometimes between ethics and science. So it's a very important part of what we do and think about all the time.
Okay, so I think to keep on time here, we'll thank Dr. Salter. And just to briefly conclude, just thank you all again for coming and listening to these inspiring talks. I hope you've learned some science, but also been inspired and been excited and think about biomedicine, clinical, scientific aspects as a career. Thanks to all of our sponsors, particularly the Gairdner Foundation, and I have the pleasure of having the title of Associate Scientific Director of the Gairdner Foundation, and I coordinate this student outreach program across the country, and it's exciting for me, as it is, I know, for the speakers to be able to interact with you people and see the excitement, and hope that excitement continues. So thank you all, thank the speakers, thank Walker for his introductions, and I'll call on Kate, who is the coordinator of this, and who none of this would happen without her. She does it all, and she's got marching instructions for you for the rest of the day's program. Thanks.